Hey, Mr. B. This is a video about operons. And operons aren't the easiest concept. You're going to want to pause, think about stuff. I try to give as many visuals so that you have the concept. They're really massively kind of simple, but really complex when it comes down to the chemistry. And so it's hard to decide what should I include, what shouldn't I include. The ultimate goal is that you understand what happened with the GFP lab, which means we need to understand the arabinose operon, and the easiest way to do that is to break it down into some smaller chunks. So this is my best attempt to do that. Let's see what happens. Okay, so first thing that you're probably wondering is, well, what's an operon? Before I even do that, I should really do a brief reminder about transcription. We're going to be talking about prokaryotes, and what the prokaryotes do is they'll have a promoter region which has the start codon and the terminator region, the stop codon, and you'll have your RNA polymerase, and what that does is unwind and unzips the DNA, and what it will do is, whoa, what happened there? Okay. Oh, I don't know what just happened there. Let's go to resume slideshow. I didn't do anything. Huh. Weird. Okay. So what it does is it makes your messenger RNA, and that it makes it on the 3' to 5' side. So what you end up with is your mRNA. It will read from 5' to 3'. Sound familiar? Okay, so an operon, what it is, it's basically, think like an engineer. It's a way to control gene expression. There's going to be times where a prokaryote wants to express genes and other times that they don't. Now, operons are in eukaryotes, but recently, they're like in the 90s where they've started finding them in eukaryotes. But for the most part, a lot of the work and the understanding comes from work with prokaryotes. So that's what I'm going to be referring to. So an operon is a group of genes that produce proteins. That's what genes do, of course. And there's two types of genes in operons, and you have a structural gene and you have a regulator gene. I'm going to show you visuals of this, okay? So just for right now, I'm just telling you there's a couple types of genes. I've got some visuals in the slide. This is where I got them from, so a lot of visuals coming up will have come from that particular site, and feel free to go to that if you want for more information. Okay, so this is something I made here. It's a concept map. Okay, what an operon does is it's a way of controlling genes, and there's two ways to do it. And there's one way called positive control, and then there's another way called negative control. And a lot of what we'll work with is this over here, but then we'll be working with this over here too, which is where it gets way more complicated for us. Just having an inducible or repressible is pretty easy. But just think about this from a control standpoint, okay? So thinking like an engineer, if we normally, what we'd like to do is break down glucose, right? We've talked about that in class. You have E. coli in your gut that, just like you, would like to prefer to break down glucose because that's easy. But sometimes you have milk, and a lot of people can drink milk. Some people can't, but a lot of people can drink milk. And as you recall from when we did our yeast respiration research, lactose doesn't break down easily, but it can break down. And so what you've got is there's going to be times where you want E. coli to be able to break down lactose. But if there's glucose, why would it do that? It would prefer to just break down glucose to make it, for its survival, for its energy. So inducible, what this means is that if I give it something, then what it's going to do is it's then going to make a protein, but so it needs something like lactose to be induced. If it didn't have it, what it has is this thing called a regulator. I'm going to show you that. And what that means, so if it's not induced, and that would just think about it kind of like normally we don't have milk in us. So normally there'd be this regulator preventing our genes that would help us break down lactose. So normally they aren't being described, so they're inactive, okay? And then there's repressible control. And normally the regulator is inactive, and so what's going on is most of the time the structural genes are being transcribed. Now, I'm kind of showing you all this stuff, and it's probably like, what? Well, let's get you some visuals, and I'll be referring back to this concept map to hopefully make this all make sense. Okay, I'll do my best. I can't promise you. This is not the easiest stuff when you're brand new to this, but okay, so remember I talked about some operons. They had genes, they had structural genes, and I'm going to circle the structural genes. They're right here. This is just a generic operon, okay? And so there should be three different genes, A, B, and C, make three different enzymes or proteins, both the same thing. And so that's what would code for so let's see what I got here. No, single mRNA. Okay. Oh, one thing I want to mention here on all these visuals. This green line, what that represents is a chunk of DNA. DNA being double-stranded, right? Now it's got the the 5' to 3' on one side, and the bottom would be 5' to 3' going the other way. So keep that in mind. This is just representing a double-stranded DNA, but it's just representing just a straight line, and this is representing a little chunk of DNA, which has a thing called the regular gene I'm going to talk about, has something else called promoter and operator, and then it has some of these other things. So this is some genes that will make enzymes. Say these are the enzymes needed to break down lactose. So that would be one example. And oh, you know what this bullet's about is one thing that's unique about prokaryotes is that you can take mRNA, messenger RNA, and it can make a big strand from these three genes all in a sequence, and then it can make multiple proteins from that. And that's kind of unique for prokaryotes. It's pretty cool. So now this regulator that I was talking about, okay, we talked about lactose, and the only time that I would want to, let's say this is for lactose and that makes enzymes to break down lactose. If I'm not drinking milk, I don't want to waste energy as E. coli to make those genes. So what I want to do is I want to have a protein right here. And this protein, what it does is it blocks the polymerase from attaching to the promoter region, which is where the RNA is going to go to, the RNA polymerase that is. That's where it's going to go to unwind and zip, and then ultimately decode or transcribe genes A, B, and C here. So this is what they call a repressor. It's a repressor protein, okay? And where that's made, which is pretty cool, is it's made upstream, and it's made at another gene, which is called the regulator gene. Okay, so again, think about it. This is all about control. And so, so this, what I'm showing here, this visual is, if there was lots of glucose present, you just had a lot of sugar, and you had like a little thing of milk. If you had lots and lots of sugar, there was going to be lots of glucose. The E. coli would prefer to break down the glucose first. Later on, once that's broken down, the lactose still being present, it will break it down, but it's going to go for the easy stuff first. So this is when there's lots of glucose present. We're not going to encode for proteins that are going to break down lactose. All right. And then I've talked about, I mentioned this thing called an inducible operon. And okay, one thing you think about programming, we want to control. Okay, program, what this stands for is promoter. You've got to have a promoter. That's where your polymerase is going to attach. You've got to have a repressor, and this is our repressor here. It's a protein. An operator, which is right here, and then the genes, which are right here. Those are the genes that we prefer to, transcribe if needed. Again, it's all about programming and controlling. So the Lac Operon is called inducible. And I'm going to come back to that concept map that I showed you. What the inducible means is that normally these structural genes are inactive, and that's because this protein is blocking polymerase. Okay, so if lactose was the only sugar present, say you hadn't had any food for a while and you pound some milk. Okay, well, you know, E. coli, they're not stupid. They're like, hey, want some food. So they're not going to die. What they're going to do is one of these genes here that I'm circling, it's really not called Lac A. It's got a different name, but I don't want to get too far in the weeds. I'm going in the weeds deep enough as it is. But what it will do is it will make an isomer of lactose, and it will make it into allolactose. And what allolactose does is it will come over to a repressor protein, and it will fit into this repressor protein. It will change its shape, and it will physically pull it off of there. So now the polymerase can find the promoter, and then it can decode. Okay, so what will happen is now our allolactose pull the repressor protein off, and that allows the polymerase now to go right along, and so we've got transcription. And so this is what we've seen on our concept map right now. It's a negative control. What I mean by negative control is that you have to take that away to then do something, right? I have to take this away to make those genes. In this case, we'll call them genes A, B, and C. And so with inducible, what that means is that normally this regulator, this is a regulator here, the regulator gene makes a protein and it pops on there. Normally the regulator is just hanging out there. So normally we're not drinking milk. And so the structural genes which are here, those aren't going to be transcribed. So now visual with this, things hopefully kind of make sense. You might need to pause, think about it, review it again. Okay. So now obviously we want to talk about this part over here, the repressible ones. And that gets into what they call the trip operon. I think that's what they say. They might say terp. But the trip stands for tryptophan. And that is a repressible operon. Tryptophan is an amino acid, one of the 20 amino acids that's needed. It's one of those basic amino acids that's needed for a lot of different cell functions. And so it's normally going to make it. So with the repressible is normally, this is what's going to happen. These are the genes that are going to make, again, they're just being generic here, call them A, B, C, D, and E. They have more complex names. You don't have to worry about that. But normally what happens, it has a regular gene, it has a promoter, it has an operator, but normally what's going on is we need tryptophan. We're using it a lot. So normally, even though this gene is made, it's made by the polymerase, it's just kind of hanging out, and it's not attached to the DNA. So normally the promoter is seeing the polymerase, and it just keeps on going, and it's just cranking along, and it's making tryptophan. So what if we had a meal that had lots of tryptophan? Do you need to keep making tryptophan? Well, no. So then what happens in this case is the tryptophan will fit into that repressor, which then is going to block the polymerase from its promoter region. I didn't keep the text on there and here. So it blocks the promoter region, which therefore now you stop making the tryptophan. So that's kind of a way of regulating itself, and they call it genetic regulation. Pretty cool. So now what we've seen here is we saw positive control. Now this is negative control. Again, negative control meaning that I have to take away the repressor to make whatever structural genes I want. So with negative control and repressible, normally the regulator is inactive. It's hanging out somewhere else. And so normally the structural genes here are being transcribed. So that's what they call active. Okay, so the words you're going to see a lot when they talk about operons, they're going to talk about, you're going to see things called inducible or repressible. Okay, so inducible meaning I need to have something to encode. Repressible meaning I need to have something present to stop decoding. It's just a little funky way of doing it, but you just work with a little bit and it will be comfortable. We're not going to do a ton with operons besides what we're doing here with the ara operon, but this is the gist of this is what genetic engineers use all the time. Is they're using operons. So far, this is what we've done. I'm just showing basically that concept map in a different way. So if this kind of setup helps you, you pause, you stop, you print this out. You look at it, what I'm showing here is the inducible operons. I need to induce negative control. I need to pull that away to allow the polymerase to encode or transcribe. And with the trip operon, it's still negative control. When it goes on, I'd have to pull that away to transcribe, but normally it's not being, it's normally not repressing because it's normally inactive, and the structural genes are normally active. So this is just a different way of reviewing. So now you've got this other part that I talked about, this positive control. I guess I didn't show the visual, but that goes back to that slide with the concept map. And what positive control is where things start getting funky. Okay, so hopefully these you can kind of follow along and you're like, yeah, that's not too bad. It's a little weird. You just got to stop and think long and hard about it at times. But positive control is where we can really get into the weeds. So what I'm going to do here is I'm going to show you a couple different videos. What I'm going to show you is just a really basic, simple explanation. I'm going to go to a video. We're going to watch a video in a video, which is pretty crazy. And so what happens with positive control is you need to have an activator. And there's going to be a protein that goes to an activator binding site, but then there's other chemicals that will bind to that, and what that does is you need to have that. So instead of taking away, you need this, which is why it's called positive. I need that to therefore to take the polymerase and do the transcription. So that's why it's called positive control. I need to attract that onto my DNA to then bring the polymerase in and do its work. So let's see this visual. We'll see this video. We'll see how it goes. I have no idea. I've included this link in case you want to watch it at your own leisure. Okay. I'll turn the volume up here. That seems like that should be pretty loud. I'm probably going to need to change it to low so it doesn't stop in the middle. I'm doing this at school. And we'll do that again. Alright. I'll make it a little bit bigger. The visuals are awesome on this.
The Lac Operon consists of a set of genes in E. coli that encode proteins for importing and breaking down lactose. The structural genes of the operon are all transcribed from a single promoter. The genes are LAC-Z, which encodes beta-galactosidase, an enzyme that breaks down lactose, LAC-Y, which encodes lactose permease, a lactose importer, and LAC-A, which encodes thiogalactoside transacetylase, whose role in lactose catabolism is not clear.
Okay, so those are the ones where I showed them just generic as being A, B, and C, and I told you that it's more complex. And yeah, so we're getting in the weeds, but generally speaking with the operons, what you have to know is what does each thing encode for. And so as a genetic engineer, if you were going to be adding on like GFP somewhere, can I get rid of all these, or do I need to keep some of these, and could I put my GFP here? Those are some of the things that you need to consider as a genetic engineer, or when you're doing some of the stuff like making and the GFP plasmid.
The preferred sugar source for E. coli is glucose rather than lactose. The genes for glucose catabolism, that is glycolysis, are always expressed. In contrast, the Lac Operon is not always on. When lactose is absent, the Lac Operon is repressed. Without lactose, the structural genes of the operon are transcribed only at extremely low levels, generating less than 10 molecules of beta-galactosidase per cell. Transcription of LAC-ZYA is repressed by the protein product of the regulator gene, LAC-I. The gene LAC-I is situated immediately upstream of LAC-CYA, but is transcribed into mRNA from a different promoter. The resulting protein, a repressor called LAC-I, is always expressed.
So one thing to point out here, there's a promoter for LAC-Z, LAC-Y, LAC-A. There's another promoter for LAC-I. It's a different promoter, and so that allows it to transcribe that at any point. It does have some self-regulation, but just want to point that out. They mention that, but it's kind of an important thing to know.
The repressor, acting as a tetramer, binds to operator regions in the DNA. One of the operator regions, called LAC-O, is just upstream of LAC-CYA and right next to the promoter for these structural genes. Another operator...
Okay, the promoter that they're talking about right there is the promoter that's going to allow the polymerase to then, basically transcribe LAC-Z, LAC-Y, LAC-A. So right now, this is blocking it. So just thinking about, okay, I talked about negative control. Right now, do you think this is negative or positive control? Do I need to take this away? Right now it's blocking the promoter, right? So that means if I had to take it away, it would be a negative control.
Region overlaps the LAC-I gene. For a tetramer to bind these two operators, the DNA loops out between them. With the repressor bound, RNA polymerase is blocked from transcribing the structural genes.
So there's your negative control. You need to take that away.
The operon is repressed. Keep in mind that even though a repressor is usually bound to the operon when lactose is absent, occasionally the repressor will fall off. RNA polymerase may be able to sneak in and transcribe the genes before another repressor binds to the operators. In this way, an extremely small number of LAC-CYA proteins can be made even when the operon is repressed. What happens if the medium is changed so that it contains lactose but no glucose? The small number of lactose permease molecules already present in the cell transport lactose inside. Beta-galactosidase then performs two functions. Primarily, it breaks down lactose into galactose and glucose, both of which can be used for energy. However, a minor function of beta-galactosidase is to isomerize lactose into a molecule called allolactose. Allolactose is an isomer of lactose, differing in its sugar-sugar linkage. Allolactose, but not lactose, is an inducer of the Lac Operon. Allolactose binds the repressor and unlocks the protein, which then releases the operator.
Okay, so now, thinking about what we just did there, think about negative control. Did we induce or did we repress? We basically took lactose and broke it down into its isomer, and that allowed it to take it away. So that's a negative control. I had to take it away, and so that looks like we had to induce it, right?
RNA polymerase can now initiate transcription of the LAC-CYA genes. When glucose is absent and lactose is present in the medium, the Lac Operon is expressed at 100-fold higher levels than without induction. The mechanism is slightly more complicated, however. In addition to the presence of the inducer, another molecule must participate in turning on the structural genes.
Okay, and where this is going is this is going to get into the positive control, the Lac Operon, and also the arabinose operon, the operon. They are both positive and negative control, which is why we kind of need to get in the weeds on this.
A transcription activator, a complex of cyclic AMP molecules and cyclic AMP receptor proteins, or CRP, must bind to the promoter region of LAC-CYA to help recruit RNA polymerase to the operon.
So what they're showing here is with this right here, and let's just call them some molecules, CAMP and CRP, and it's just a protein. Without that, your polymerase cannot transcribe LAC-C, LAC-Y, LAC-A. It needs that. And so then they'll talk about how that's made here. So listen carefully. So you're probably wondering, well, what the heck? How's that there? And I'll talk about that right now.
What happens if, in addition to lactose, the medium also contains glucose? In this case, the operon becomes repressed because cyclic AMP levels in the cell plummet, leaving the activators non-functional.
Okay, so that's why it's positive control. It needed those on the promoter region, right? So that's why it needed it. If it needs to have it there, then that makes it positive control. And what happens when glucose is present, the cyclic AMP and that cyclic AMP protein, they drop down dramatically. So they have an inverse relationship to glucose. Low glucose, high cyclic AMP. So that's low glucose, high cyclic AMP. High glucose, low cyclic AMP. So that makes it the positive control part of this.
Glucose inhibits cyclic AMP production by a mechanism that ties the synthesis of cyclic AMP to the transport of glucose into the cell. That is, if glucose is transported, cyclic AMP levels fall. When E. coli cells grow in a culture medium containing both glucose and lactose.
Okay, so this is like you had sugar plus you had some milk for something for lunch.
The growth rate of the population varies over time. During the first burst of growth, the cells use only glucose. After depleting glucose, the growth of the population stalls as the cells induce the Lac Operon. During the second burst of growth, the cells use lactose. The term catabolite repression describes the choice of nutrient use during the first growth burst. Here, the presence of glucose, a more favorable catabolite than lactose for the cell's energy needs, has essentially repressed the Lac Operon.
Alright, so let's go back to the PowerPoint slideshow. Alright, so that has a lot more meaning than this stuff, right? Is that the same one? It is. Yeah, so what I'm going to do is make a separate video for the Operon. I've got to think about this. What I might do is just discuss the Operon in class. But we do have the 2-hour delay thing that I got to work into my brain, so I'll come up with something. But this is Operons. It's heavy duty. You might need to watch it again. Definitely if you're interested in doing some kind of genetics type stuff, having a basic concept of operons. And what I mean by that is if you could really put together this concept map and make sense of this concept map by looking at visuals and think of a visual and know when is it positive control, when is it negative control, what is inducible, what does repressible mean. If you can do that, and that's why I've got, these are really basic visuals, they're not that fancy, you know, obviously there's lots of videos on YouTube, some are great, some are interesting, some are very weird. I've looked at a lot, so trust me, but if you can kind of get this concept down, you will really knock the socks off of your professors in college going in as a lowly freshman or maybe a sophomore. You'd really impress them because you'll have a leg up. But there's way more information on these. I'm just scratching the surface, believe it or not. Okay, well, so sorry about the long video, but it is what it is. Thanks.